together and asks you what's the likelihood of getting this phenotype in the progeny. You don't have to figure out all of the possibilities, right? You can always use probability just to figure that out without figuring out, okay, here's all of the progeny I can get. You can just ask, what's the likelihood of getting this combination? Just like question number 2 on the genetic concept, when they say, all right, here's the parents, what's the likelihood of getting this one? You don't have to figure out all of the possibilities to be able to answer that question. Okay, so we're going to go on today and talk about maybe some things that you probably touched on with Biology 160, but they're kind of the more interesting things or strange things about genetics that don't follow exactly the Mendelian ratio. So when he worked with worms, he was lucky because all the traits he picked worked perfectly. They weren't linked genes, they weren't on sex chromosomes. So it was very straightforward for him to figure those patterns out. But then later on, as people continued studying, they kept finding these strange inheritance patterns that didn't seem to fit. So we're going to talk about X-linked and Y-linked or sex-linked inheritance. Some variations on dominance patterns. So that we'll talk about incomplete dominance and codominance. And then epistasis explains this right here. So lab coat color can actually be explained by gene interactions, where one gene can actually mask whether or not you see the trait encoded by the other gene. And then we'll talk about penetrance and expressivity. Sometimes you have an allele, but it doesn't get expressed. So, for example, we talked about polydactyly yesterday, having extra digits. And with that, some people who have that gene have variance on how severe it is. It could be a fully formed extra digit, or it could just be a skin tag that's not that noticeable. So that's variation on expressivity, but also sometimes there's variation on penetrance, where you actually might have this gene, but you don't have the trait that goes with the gene. And then the last topic actually fits perfectly with the genetics discussion, which is how do we really parse out how much genetics influences traits versus environment? And this is the age-old nature versus nurture debate. And some of our traits are really kind of fall into that category. All right, so sex-linked inheritance is complicated because of differences between males and females. So we know that males have an X and a Y, at least in humans, right? That sex determination, it's determined by, there's a locus on the Y chromosome. It's called SRY gene. The SRY, essentially, during development, if an organism doesn't have SRY, the default is to go into the female development. So it will follow the pathway to become a female unless there is SRY. If SRY is present, it will then develop male sex characteristics instead of female sex characteristics. So the presence of the Y for mammals determines sex. The X chromosome, we both have, females and males have X chromosomes, and in fact, the genes on the X chromosome, they do a lot of different things. It's not specific for one sex or the other. So we talked about colorblindness. This is the colorblind test, right? And that gene that encodes the ability to distinguish color is on the X chromosome. There's, I think, something like 2,000 genes on the X chromosome. So there's a lot of traits that are X-linked. And with X-linked traits, because males just get one copy of an X, they don't have a backup copy, so they tend to have more of the X-linked disorders or traits that are associated with the X chromosome. The Y chromosome, I don't know if you've ever seen the picture of the Y, but the X is just kind of a normal size. The Y is a tiny little stub. It's a very small chromosome. There's actually few Y-linked genes. Kind of makes sense because the genes that are on there are important for differentiation into becoming a male, and females don't have those genes, so there's few Y-linked traits or Y-linked disorders. Mostly when we talk about sex-linked, it'll be X-linked. We talked about why men are more susceptible. The other term I want to introduce is this one. So we've already talked about homozygous and heterozygous before. Hemizygous is what we use to refer to a male because they only get that one copy, the one half. So hemi is like half, right? They only get one single copy. Then a couple of notes about nomenclature. I do notice sometimes people, when they write X-linked, I see lots of different ways that people write the genes. The convention is always to use a superscript with the letter. So for every, if it's an X-linked gene, the gene is on the X chromosome, you write the X chromosome and then you use the letter. So if we were doing color blindness or something like that, we would use a C. If you have an unaffected allele, you still put a letter. So that's another thing. Sometimes people just would leave off the letter because it's the wild type, but you still put a letter. It's capital or lowercase. And the Y does not have a superscript because it does not carry the gene at all. That same gene is not on the Y. So you put nothing on the Y, you just put it on the X. All right. So yeah, we talked about some of these. Color blindness, hemophilia is another one, and Duchenne muscular dystrophy was one we had talked about earlier this quarter. Okay, so one common example of this, and this, again, we're going back to a royal family, because royal families do have a lot of, they keep it in the family, so things kind of stay within that family. But a very famous example of hemophilia was Queen Elizabeth was a carrier. So if she's a carrier, she married a male who was phenotypically normal. So I want you to try to write out what that cross would be between Queen Elizabeth and her husband. If she's a carrier and he is phenotypically normal. And so what would be the likelihood that they would have affected sons? And what would be the likelihood that they would have affected daughters? So first step is to figure out what are their genotypes, right? So for heterozygous female, you have X big H, X little h, and then a phenotypically unaffected male would be X big H, Y. I want to actually remove normal out of all of my stuff. I didn't fix this one yet. So the cross would be this. So looking at this cross, you set up the Punnett square exactly like any other Punnett square, right? For their female daughters, so for their daughters, how many of them would be affected from that Punnett? So I guess affected, they'd have to have two, they'd have to have X little h, X little h. So they're not going to have any, their daughters shouldn't be affected, but for their sons, how many would be affected statistically? Half of them, right? Half should be affected, half should be unaffected. For the females, half will be unaffected, not carriers, and then there would be unaffected carriers, because this is a recessive trait that you have to have two copies of the X little h. So if we were to give the genotypic ratio, this is, the X-linked ones become more complicated when you write out the ratios, because you have to write out all the possibilities. So that would be 1 to 1 to 1 to 1. And make sure when you write it, you don't just write the numbers, you always like include what are the genotypes. And the phenotypic ratios, oh, again, I have to fix this. Unaffected, one unaffected female, one unaffected carrier female, and then unaffected male, and one affected male. Okay, so does that make sense to everybody how to do that? I think the problem that you have, I'm going to look at it again, is it's also, oh, it's a Duchenne muscular dystrophy. So everybody take a look at that, and let's see if we can answer number 5. Maybe discuss also, that's a good one to think about. So look at number 5 and then see, and we'll talk about what the answer should be for that one.
When it says there's no history of DLD, does that mean absolutely none?
Yeah.
Like we didn't have any boys, so we didn't know.
Okay, so can someone read the problem out loud? So we can see if we can draw out the,
Red-green
okay.
color blindness is an X-linked recessive trait. Betty and Timothy have 5 children, 3 boys and 2 girls. 2 of the boys and 1 of the girls are colorblind. Timothy is red-green colorblind and Betty has normal color vision. What are the probable genotypes of Timothy, Betty, and their children?
Okay. So, yeah, I guess, was I reading it differently? I was reading number 5.
Oh,
Okay.
just kidding.
Okay. Well, that, sorry, was everyone reading number 4?
No, I just did
Number 5.
Number 5?
Yeah.
Okay, do you want to read number 5?
Sure. Duchenne muscular dystrophy is caused by an X-linked recessive allele. An unaffected man whose brother has DMD wants to have children. There's no history of DMD in his wife's family. What is the probability that the first child will have DMD? Explain your answer.
Okay, so we have an unaffected male. He himself is not affected. So we'll use D for Duchenne muscular dystrophy. If it is recessive, the little d would be the affected allele, right? So what would be the man's genotype? This man in the story problem, not his brother.
X capital DY.
Yeah, so he is unaffected. So he does not carry that allele. His wife, it says there's no family history at all. So we're going to assume that the only way for this to arise would be through a spontaneous mutation. But we're going to assume that she doesn't have it or carry it in her family. So hers, presumably, what would you assume her genotype to be?
Capital B's.
Big B, right? We assume that since nobody has ever in her family had it, she probably is not a carrier. His brother, though, so I'm going to show you the pedigree style here. When you have branches like this, you have siblings. So what would be, and this is the symbol for males, his brother was affected, or color in his brother. So what would have been his brother's genotype?
X little d.
X little dy. So should this couple be concerned about their children?
Not at
What
all.
do you think? Everybody thinks no? So you say, what would be the likelihood? What number did you put? 0, right? We're going to assume 0 because we're assuming that there's no spontaneous mutation. So since he's not affected, that's the key information there, right? Now, if you had let's say that so you have the answer for that question, but let's say that it was on the other side. Let's say that we don't know what this female is. We do know that she had a brother. So I'm going to take the brother and put him on this other side. Let's say that her brother was affected. Don't know her genotype. Should this couple be worried? Like the man side has no history, the female side, she's actually has a brother. So what would that tell you about the parents if her brother was? Yeah.
The mother is at least a carrier.
Yeah, so we know, how do you know it's the mom and not the dad?
Because the X chromosome comes from the mother and the male.
Yeah, so we know that this son is going to get his X from his mom. He gets the Y from his dad. So the dad is unaffected. The mom we know has to be a carrier. We know she's a carrier because this sister is unaffected. So, and also they did post that she's affected. So we're assuming that we don't know, right? She could be basically 50-50 chance that she's a carrier. So in this family, you'd probably recommend testing. If they were really interested in having a child, they could both get tested. And if she knew she was a carrier, then there would be options for them. They wanted to do like pre-implantation genetic diagnosis where they select an embryo. But anyway, they should be concerned. Whereas on the other side, if it happened on the other side, there wouldn't be any concern. Okay, so any questions on X-linked traits? All right, I'm gonna skip over that one. Okay, so now we're gonna move on to dominance. Now, most of what we have talked about, we said there's either dominant or recessive. If a trait is dominant, how do you know that it is dominant? What is kind of the easiest way to test to see if it's dominant?
Check the generations.
Okay, so you could check generation, so a little bit more information, so how do you set up that cross? Yeah, definitely you want to add.
You breed two homozygous of different traits, and then whichever one is dominant will appear in the progeny.
Okay, so that's getting at the core of the definition of dominance, is that the dominant trait shows up in the heterozygote. So dominant allele is expressed in both homozygous condition and heterozygous. Okay, so that's our classic definition. Well, it turns out there's a lot of variance on that. Now, we talked about explanation for why something would be dominant versus recessive at a molecular level usually has to do with the protein that is encoded, right? And usually the dominant either is more active or it's expressed at a higher level, or we talked about all those explanations. Sometimes one allele is not dominant over the other. So in this example, codominance occurs when both alleles are equally expressed. So the traits encoded by both alleles are expressed. So you could say for this definition here, codominance occurs when the traits encoded by both alleles are expressed to the same extent. So we see both. And the classic example that I think every genetic textbook gives is blood type. So blood type, so ABO, does everybody know their blood type? You might also know the plus if you, the RH factor, your plus. But the blood type is encoded by this gene called the I gene. And the I gene encodes a transfer enzyme that puts a sugar group on your proteins. So you have glycoproteins. This enzyme transfers a specific sugar group to the very end. So you can see that everybody has the base for this glycoprotein, but depending on whether you transfer, I forget what that end sugar is. One's like a galactose and something else. But the sugar that adds either the red or the blue here, that specific sugar determines whether you have type A or type B. Now, if you have IA, IB, one of each, the enzyme is half, these enzymes are putting the A sugar group on. These enzymes are putting the B sugar group, so you end up with AB. You have both of those on your red blood cells, okay? How do we get O, type O? Does anybody know what happens for type O? It's shown right here with the little I. Yeah, it's basically a broken enzyme, so it's not transferring anything. So if you have little I, it means that there's no additional sugar that gets passed. If a person has two little I's, there's nothing extra added to the base. So you see there's no red or blue. They would have type O. This person would have type AB. For type A or for type B, there's actually two possibilities. Right, so if you have type A, what are the two different ways what genotypes could you have?
Little I,
Yeah, so you could have both IA, or you can have IA, little I. And the same is true for B. So there's two ways to get type B blood. Now, used to be that people would use blood typing as a way to test for paternity. There's some interesting things that can happen, though, with epistasis, and I'll give you an example of that. So this is not the most reliable way to test for paternity, as we know. DNA fingerprinting is much more reliable than blood typing, but historically, this was a way that people used to test. So I have this question here. I actually think this might be, let's do this problem, and then there's another problem very similar with blood type in the problem set. So a male whose blood group is type A and a woman whose blood group is type B have a son who is type O. So what do the parents have to have? If they have a type O, yeah, they have to, both of them have to have a little I. So they have to be IAI, little I, and then IB, little I. What is the probability, if these are the parents, what's the probability of the couple's next child being type B? So So to figure that out, you just have to make a little Punnett square. And again, Punnett squares are exactly the same setup. We just include the superscripts. So what is the probability that they would have type B? Can anybody see from that Punnett square what it would be? Yeah. Would you say 25? Yeah. Everybody see that? 25%? What chance would they have of having an IAIB? 25%. Right. Okay. So in the problem set, there's another one like that. It basically gives you the genotypes for the parents, and then you have to figure out what's the likelihood for each of the blood types. Anybody have questions on how to do that one? Okay. So we will continue along so I guess actually let me add to our definitions here as we're going. So if we said a dominant allele, if it's completely dominant, is expressed in both homozygous and heterozygous, what would you say for codominance? Codominance, what happens in the heterozygote that's different than with complete dominance? So I'll say heterozygote, what? Expresses both of the traits. So expresses both traits. Okay, that's codominance. And then the last variation that we'll talk about is incomplete dominance. And this was first noticed with flowers. And there's a lot of examples of flowers that have incomplete dominance. And you'll see when you understand the molecular explanation why that makes sense. But this is for 4 o'clock flowers. It's also true with snapdragons. But if you have, let's say that you have a red and a white flower, true breeding, so they're homozygous, and you cross them together, in the hybrid, you get 100% pink, which is not red and it's not white. It's something intermediate between the two parents. Now, I want you to think about how could we molecularly, at a molecular level, explain what is happening with incomplete dominance, where you're getting something in between two. And let's just tell you that the enzyme that's encoded by this gene makes a pigment. So what do you think happens in the heterozygote? Maybe you can talk to the people next to you, try to figure out how do you explain that at a molecular level? How you get something in the middle.
Started with one.
I don't know. Oh, probably because they have that thing in, right? So that would work. I mean, that could be true. I don't know. Maybe it's like mixing colors. I have no clue.
Yeah.
I mean, like, because they have like one gene, right? And then they have the other. So that gene probably worked a little bit. Like they have half of it.
Okay, so what would you say? How do you explain the pink coming from red and white? Yeah.
I would say that maybe the dominant allele doesn't completely mask the recessive, which is why part of it shows.
Yeah, so that's a good idea that it's not enough to have half of the amount of that enzyme. So dosage matters here. Anyone want to add anything else to that? That's kind of the basic idea, is that the heterozygote dosage matters to get the complete phenotype, so that's why you don't get red. I like to think of this in terms of baking, so I bake a lot of cakes for my kids and stuff, and if you think about each of those genes as supplying a certain number of drops of color, like if we're talking about pigment gene, let's say that big R can give you 5 drops of red color. So if you have a homozygote, you're getting 10 drops of color. But if you have a heterozygote, the little r is a recessive allele. It's a broken enzyme, so it's not adding anything. Here, you're only getting 5 drops. And 5 drops in your frosting will look different than 10 drops in your frosting. So that's kind of how I like to think about it. If you then take your heterozygotes, cross them together to get your F2 cross, you're gonna get now 3 colors. You get the full dosage, big R, big R. Your heterozygotes are gonna be pink, and then the little r is gonna be white. So most of the examples with this one, with incomplete dominance, do tend to have to do with like pigmentation or other things where the dosage of the product matters for the phenotype. Okay, so I don't think there's a problem on there, but with that. Okay, so let's look at all of the dominance definitions. Complete dominance. So the definition says phenotype of the heterozygote is blank as the phenotype of one of the homozygotes. What would you think should go on the block? The blank is the same. So phenotype of the het is the same as the phenotype of one of the homozygotes. That would be complete dominance. That means that dosage doesn't matter. As long as you have one copy of functioning protein, it's enough to give you the phenotype. Incomplete dominance, phenotype of the heterozygote is blank the phenotypes of the two. Mixed, you could say intermediate, between, or is in between the phenotypes of the two homozygotes. It's different, right? So now you have a new phenotypic class. And then codominance, phenotype of the heterozygote. Oh, I guess, I don't know what I intended to have here, but expresses the phenotypes of both homozygotes, or, yeah, both alleles are expressed in that one. What did I, oh, yeah, I said includes the phenotypes of both of the homozygotes. All right, so everybody have a good understanding of these? Okay. So our last topic, or not our last topic, but one of our last topics, is about epistasis. I think this one is really fascinating because it kind of shows you how complex genetics is. Genetics is typically not about just one gene controlling one trait. And we always talk about those examples because that makes it easier to understand inheritance patterns. But for a lot of our genes, they're operating in pathways with lots of genes. And so there tends to be gene interactions. So epistasis is this phenomena that you can have interactions between two genes, two or more genes, where one of those alleles has the ability to hide or to mask another gene. So the best example that I know of that's easy to understand is with lab coat color. So if you're familiar with labs, you know that you can get a black lab, chocolate lab, or a yellow lab, right? And this is determined by pigmentation genes that they have. There's two genes involved in the coloring here. The one is called the E gene, and the other one is called the B gene. So these are the two players in this pathway. So to kind of explain molecular level, well actually before I explain the molecular, I'll just tell you, if you have at least one big B and you have at least one big E allele, you will get a black lab. If you have little b, but you have at least one big E, you get a chocolate or a brown. And then if it's little b, or it can be actually anything right here, but if you have little e, it always turns out yellow. So which one of these genes seems to be the masking gene that can hide what is at the other locus? Yeah, this one. This is determining whether or not I know what's at this locus. As long as it's little e, it will always look yellow, and I will have no idea what's over here. This could be this, or this. But as long as it has little e, it's going to show up as a yellow lab. So in this, E is the epistatic gene, meaning it is the gene that hides the other gene. So to give you sort of the explanation for this, molecular explanation, again, it's pigmentation pathway, so we're talking about production of pigment. And the E gene produces this protein that makes eumelanin, which eumelanin is what gives you that dark color. The B protein controls how densely deposited that color is. So if you have big B, you are densely depositing pigment, so it makes sense it's going to be black. If you have little b, you still make the pigment, so you're producing eumelanin, but you're not depositing it as densely, so then it appears brown. And then if you have little e, so you don't even make eumelanin at all, you actually produce an alternative pigment, which is called pheomelanin, which is the yellowish color. Pheomelanin can also produce like a reddish color as well. But pheomelanin is deposited instead of eumelanin, and that is why you always will see yellow, because this happens upstream in the pathway. And so first you have to be able to make the eumelanin pigment, and then then you can see how densely you deposit it. But if you don't make eumelanin, you're going to turn out yellow. Okay, does that make sense to everybody? So there are some questions. I think there's one question about with dog coat color on there. Let me see what the question is. All right, so it explains how it's determined. And the first question is which of these loci, that's just talking about which gene is epistatic. And then it asks you to write out all the possible phenotypes. So I think that should be simple enough. The last question is saying that if you cross a black lab to a yellow lab, what do you get in the F1? And then determine the phenotypic ratio of the F2 if you cross the F1 from that cross to a homozygous recessive dog. So let's maybe look at number E briefly here. So try to figure out for that first description of the cross, if you're crossing, this is what it said. Big B, crossed with this. Can anybody see just by looking at it, what would the progeny look like? Or what would their genotypes be? I think you said it now. All heterozygous. Yeah, it's going to be all heterozygous. So, make sure that you know how to, we would figure out the gametes from this, but actually it turns out when I do first and outside, inside, and last, it turns out they're all four the same, right? So the only gamete that this one can produce is big E, little e. The only gamete that this one can produce is little b, little e. So all of the progeny are going to look like this, right? So that would be your F1. What color would your F1 progeny be? What color would those dogs be? Black. You'd have 100% black in your first generation. Then the cross that they actually want you to solve is to take one of these, and it's a back cross. Cross it back to this one, which is little b, little e, little e, little e. So then you can either use pork leg method if you want, or you could do a Punnett square. It's not a typical dihybrid. You'd have to figure out what are the possible gametes. But can maybe take a minute or two to make sure you know how to do that one. one, does anybody have numbers that they wanna feel confident about? Yeah, you wanna share what you got?
1/4 black, 1/4 brown, 1/2 yellow.
Do the others get that number? Does that sound right? So let's look at, just to make sure you kind of have this cross set up. So if these are the parents, right, for this one, there are, we can look at all of the possible gametes that they can create. We do the B little b, the B little e. Figure out their gametes, and then FOIL, figure that out. So we're gonna do first. And then outsides, the insides, and then the lasts. So those are the 4 different gamete combinations. This one though, because they're homozygous recessive, there's only one gamete that they can produce. What is their one single gamete? Little b, little e. So you could write that out 4 times, or because we know that it's going to be 4, we don't have to write all of those out. If you want to simplify your Punnett square, you can just do that.
If it's small, just
And you know that all of these are the same. So when we combine these, we get these combinations. Little b, little b, big E, little e.
This one or that one? Either way.
Okay? Then we just have to assign phenotypes. These should be 25% of each, but then we figure out what color would they be. So this one, what color would that be? Black. Everybody see that that one is black? This one has little e, so what color is it?
Yellow.
This is yellow. This one is little b, but it does have one big E.
Brown.
That one's brown. And then this is little b, little e,
Yellow.
so that's gotta be yellow. So we have 50% yellow, 25% are black, 25% are brown. So if you think about this, this is important if you were a dog breeder, right? If you're trying to breed like chocolate labs, or you're trying to breed yellow labs, you have to think about the genotypes of the parents and the process that you're setting, right? Okay, any questions on that one? Okay. So, I have one more example. I like to throw this in because it's actually based on a true story that happened with blood typing. So there was a male that had type AB blood, and then had this female who was type A blood, and they discovered that their baby had type O blood. And then the man becomes furious with the wife because he says, this baby can't be mine. So is it possible, if his blood type was AB, her blood type was A, is it possible for them to have a baby with type O? Not based on what we know, right? Why is it that this is not possible? Yeah.
He himself would have to have a little I
In order to have that. So this actually did happen. This happened in India. They call this phenotype the Bombay phenotype because this is a true story, but the baby actually was his. It turns out that in addition to the I gene, there's another gene that controls the stem, basically the part of when you're building the glycoprotein. Let me go back to that picture of the blood group. So the I gene controls what sugar you add, but there is a gene called H gene that controls this little stem that starts off the whole process. If you have little h, this basically is incapable of building the stem to attach the sugars onto. So you could have little h, and then you could have IA, IB, but in the blood cell it wouldn't attach anything because there's no way to hook it on. So yes, you could have IAIB, but you would never know that, and you would appear type O, because when they do the test for blood type, they're just testing for whether or not you have the A antigen or the B antigen. So this is another example. Can anybody see what this is an example of? One thing is, yeah, epistasis. This is another example. Which gene is epistatic? Which one hides the other one? H. So the H gene is the epistatic gene to control whether or not you can see the I gene. What this means is that those parents, that coming back to the two parents that we had here, one was IAIB and one was type A. We don't know the genotype. What do we know about their H locus? What would they both have to have in order to have this child that appears to be type O? Yeah, both of those parents had to be H, so that they could have a child that got little h, and then appeared to be type O. So this is one other example of epistasis. Okay, I just have a few more slides. I think I'll save pedigree stuff for tomorrow, so we'll just talk about penetrance and expressivity, and then we can do the worm stuff, and then the last part of class we can do a discussion. Okay, so penetrance and expressivity are even stranger, where sometimes people have an allele, but they don't express the phenotype that goes with that allele. The example I like to give, or this visual I think helps to explain it, where if you had big B, and you cross it with little b, all of the progeny are heterozygous, right? 100% of the progeny should have some phenotype. If it was complete dominance, what color would you expect them to be?
Purple.
Fully purple, right? Just like this one. If it was complete dominance, if it was, let's say it was incomplete dominance, what color should all of the big B, little b look like? Like a light purple, lavender, somewhere in between those two. So what actually happens with penetrance, with variable penetrance, is that even though all of these individuals have the same genotype, they are all big B, little b, some of them have it and some of them don't. Well, that seems kind of random. Like, why would you have that gene but you don't express the trait? And then with expressivity, they all have a little bit of the trait at least, but some of them have the very severe genotype. They have the full color. Some of them just have a little bit of the color. So that's called variable expressivity, where they kind of show that with different shades. Variable penetrance and expressivity means that some people, they're big B, little b, but they don't show any of the symptoms or the trait. Some people have the full range, some people have something in between. Now imagine if you're studying a disease or a disorder and it has variable penetrance and expressivity, and you're trying to make a family tree and track down how is this being inherited. You can see how confusing this is, because even when you're supposed to see something in an individual that has this particular genotype, you don't see it. So this makes genetics really complicated. I have a couple of examples. One that we've talked about a little bit in class was polydactyly. Polydactyly it's dominant. Remember that it's dominant. So if it's dominant, what does that mean should happen? How many alleles do you need to have to express it? You just need the one copy, and you should have polydactyly. But what actually happens is that some people who have that trait, they don't even express it at all. So there's variable penetrance, that they may carry that dominant allele, but they don't have it. Expressivity means that sometimes, as I mentioned, you can have a range. It could be a very severe phenotype with a fully formed extra digit, or it could be a skin tag, which is very minor, right? Can anybody think of thinking on a molecular level, how do you explain why there might be this kind of range of expression of traits that you should have that? I mean, you have the gene that encodes that trait, but why might it, in different individuals, why might it show up in some and not show up in others? Anybody think about, and kind of go back and think about epistasis and think about the complex individual.
Would it have like an additional pathway that affects the expression? Or?
Yeah, so a lot of these things are a part of a pathway. There's lots of genes, and we know what that person's genotype is for that one gene, but maybe in the background of their genetic background of all the other proteins that are part of the pathway, that can create a different scenario at a molecular level so that you don't have the same developmental patterning effect, right? So these are usually explained by gene interactions with multiple different genes, and it's hard for us to know like what that individual has at all these other loci. If we're just tracking the one polydactyly gene that we know of, we don't know about all the rest of it. So that always makes genetics complicated when you're studying a trait. And we want to be able to pin it down on one thing, but you usually can't. Okay, one other example. I love dogs. I used to have a dog that was a Rhodesian Ridgeback mixed with the Walker Coonhound. And the Walker Coonhounds have these tricolor patterns, but my dog actually looked like a Rhodesian Ridgeback. She didn't have the coloring of the, well, also the Beagle has the tricolor. But this is, this gene SP encodes the spotting color, and you can see that dogs can have variance in how much spotting they have. So it shows you a range of different phenotypes that you could get. They all could carry that same exact allele, but then they all look different. And again, the explanation is the same, that these kind of pigmentation genes and stuff, they're part of a pathway. So that can affect how you get the variation. All right, anybody have any questions on the terminology? I think for these two, you just want to be familiar with what does it mean to be variable or to have variable penetrance and variable expressivity, that you could identify if one of those phenomena is going on. All right, so we can take our break, and then when you come back, you can do your scoring of your paralysis. And then the very last, I think starting at like 1 o'clock or however long it takes people to score, then we will start our discussion. After you're done with the plates today, all of your stuff can go into the bucket. So we're all done with the work. So say goodbye to the work. We're all done.
Oops.